I am Jesse Isaacson in Prentice's AP biology class, and we did a lab where we injected foreign DNA into E. coli bacteria. So to start out, we took some E. coli from that was provided to us, and we spread it around on this plate so the bacteria could grow and we could get fresh new colonies the next day. And the next day we took some of those colonies, we put them in vials filled with calcium chloride solutions. The calcium chloride increases membrane permeability so they can take the foreign DNA better. We put one colony in 3 different vials. One vial had kanamycin resistant plasmid. Kanamycin is an antibiotic that will kill bacteria. Another vial had ampicillin resistant plasmid in it, and ampicillin is another antibiotic. That plasmid also had a segment of DNA called GFP. GFP comes from a jellyfish and causes the jellyfish to fluoresce. So hopefully the plasmid will make the bacteria fluoresce too. Then in the other vial we had nothing. It served as our control group. So we put the bacteria in the vials, we chilled them, we heated them, and we chilled them again. And this sequence helps to transfer the plasmid into the bacteria. Then we added broth, which is their food, and we mixed it. We spread this bacteria broth onto each of these 6 plates, and we put beads on them, rolled it around to cover the bacteria, to cover the plate with the bacteria. And these 2 plates, they are just broth and with no antibiotic in it. This one has the ampicillin and GFP bacteria plasmid. This one has the kanamycin plasmid bacteria, and you can see the bacteria is everywhere. It's covered completely. In these 2 plates, are kanamycin plates with the antibiotic on it. This one was just the E. coli with no plasmid in it, and you can see the antibiotic killed all of them. This one has the kanamycin plasmid in the bacteria, and you can see there's a difference between these 2. There's a lot more bacteria on this one than this one. It's because only some of the bacteria got the plasmid and the rest died. And then these 2 plates have the ampicillin in the agar, which is what they reside on, and this one has the GFP ampicillin plasmid in the bacteria, and you can see that they formed a bunch of colonies, but not as much as the one with no ampicillin in it. And this one was just E. coli with no plasmid, and they all died. You can see a few specks in there. That is not E. coli. Ampicillin breaks down after a while, so the foreign bacteria can grow on the agar. That's not E. coli. And you notice a difference between these 2. This one is slightly green and this one is not. That is because the bacteria on this plate does not need the plasmid because there is no ampicillin on it, and whatever life does not need, it will do away with, otherwise it's just wasting energy. This one needed the ampicillin resistant plasmid in order to survive, and so it also has the GFP, and that is why they are green. Okay, so the plate on my right is the kanamycin transformed bacteria, and the plate on my left is the AMP and GFP transformed bacteria. And you can see that the one on my left is glowing and the one on my right is not. That is because the GFP segment of DNA in the plasmid causes the bacteria to glow. It came from a jellyfish, and the jellyfish produces a protein that these bacteria now have, and now it glows. The other one does not.